Good morning, students. Now let us discuss about a different concept in genetics and a new concept and a very advanced concept, that is linkage and recombination. Now let us understand what is the meaning of linkage and what is recombination. So now linkage is nothing but this concept which is not explained by Mendel. Mendel has conducted so many experiments. Mendel has given basic idea for genetics, but he could not know about these two concepts, and which is explained, which is understood by the later scientists after the death of the Mendel. Now let us understand what is the meaning of this linkage. Linkage is nothing but when we take a chromosome, okay, different large number of genes present on the chromosome. Okay, on the same chromosome, different genes are present. So that, and these genes which are physically associated with one another, so that the physical association of genes on the same chromosome, now this is the same chromosome and so many genes are distributed, and there is a physical association between different genes which are located or which are situated on the same chromosome is called as a linkage. For example, now this is one chromosome. Suppose if there is one more chromosome and genes are present on this chromosome also. Now when the genes present on this particular chromosome and genes present on the another chromosome, these two are not linked. Suppose if these genes, when they are present on the same chromosome, those genes are called as linked genes, and the phenomena is known as linkage. And when crossing over takes place, you know that very well, the process of meiosis. During the process of meiosis, crossing over takes place between non-sister chromatids. So say for example, now this is one chromatid and this is another chromatid, and this is one particular chromosome and this is another chromosome. So this chromatid is one, this is another one, and these two are non-sister chromatids. Between non-sister chromatids, sometimes what happens, crossing over takes place. So during this process of crossing over, and there is a non-parental combination of genes, non-parental gene combination is generated. That process of generation of non-parental gene combination is called as a recombination. Say for example, just I can give an example. Now this is, say for example, this is a chromosome and we have got a genes like capital A, capital B, capital C, capital D are the genes. There is one more chromosome, and on this the genes are small a, small b, small c, small d genes are present. Now what happens here? Okay, crossing over takes place. When crossover takes place, okay, crossing over takes place here like this. Crossing over takes place. So here what happens? It results in the non-parental combination takes place. Capital A, small b, small c, small d. So now this capital A, small b, this is non-parental combination of genes. So the process of generation of a non-parental gene combination is called as a recombination. This was the concept which is understood later after the death of the Mendel. Mendel could not know about this linkage and recombination. So these two concepts which were understood by the later scientists after the death of the Mendel. Now this linkage and recombination, this process of linkage and recombination was understood by different scientists after conducting so many experiments, and T.H. Morgan, Thomas Hunt Morgan, you know that very well, who has experimentally verified the chromosomal theory of inheritance. He proved afterwards by conducting experiments by selecting an organism known as Drosophila melanogaster. So here T.H. Morgan has selected Drosophila melanogaster is a fruit fly, commonly known as fruit fly, and a simple fly which he could select for his experiment and to study the genetic characteristics. You know that very well. This was selected by the Morgan because of so many other reasons which we have discussed already in the previous class or in the previous video. Now T.H. Morgan, he conducted experiments. Okay, how he has conducted experiments to explain the process of linkage and recombination. So Morgan he conducted several dihybrid cross experiments. He conducted several dihybrid crosses in a Drosophila melanogaster to study the sex-linked genes. To study the inheritance of a sex-linked genes, he conducted this experiment. So now let us understand the typical concept here. You must know about some inner idea which is present behind this. Okay, here to know the sex-linked genes, inheritance of a sex-linked genes, how the genes which are linked with the sex chromosomes, genes which are present on the sex chromosome or particularly on the X chromosome, how it gets inherited to the next generation. To understand this, Morgan conducted several dihybrid cross experiments in Drosophila melanogaster. So while conducting this Drosophila melanogaster, you know that very well, it has got many genetic variations, okay, in which Morgan, he hybridized between, he has selected some characteristics to conduct this dihybrid cross. Like Mendel, he conducted a dihybrid cross. What Mendel has taken characteristics like round shape and yellow color is crossed with wrinkled shape and green color. In the same way, in a Drosophila, in fruit fly, he crossed with, this is a male individual, brown body, shape of the body, brown body, color of the body, and color of the eye. Here brown color of the body, and this is color of the eye. So these two characteristics are considered in male individual. This male is crossed with another female having yellow body with white eye. So now he considered this is wild type or dominant one, and this is a mutant type he considered. He said, so he considered it as a dominant one, and he considered it as a recessive one, or otherwise it is a wild type. Generally we will use the word wild type, and it is a mutant. So when he crossed, okay, and afterwards, okay, later when he got F1 individuals, and these F1 individuals are F1 progenies, he intercrossed. You know that very well. Mendel also, after getting F1 generation, and all the F1 plants were self-pollinated. In the same way, he intercrossed. What Mendel self-pollinated, in the same way intercrossed the F1 progeny. And after crossing F1 individuals, intercrossing F1 individuals, okay, what the Morgan expected, he could get a result like Mendel. Okay, but he got a different result in the examination. Sometimes they may ask what was the result of a T.H. Morgan after conducting this hybridization experiment in a Drosophila melanogaster. So now let us see what result he got. And these genes, brown body and red-eyed, they did not separate independent of each other. So according to Mendel, so that he has taken two characteristics like shape of the seed, round seed and yellow color. He has taken these two characteristics, and these two genes were separated according to Mendel. But this is what he was verifying. But these two characteristics, genes for these characteristics, did not separate independent of each other. This was stunning result for this Morgan. And afterwards in the F2 ratio, so this was after the F1 generation, when he intercrossed, he got this result. And in the F2 ratio, what Mendel got in his experiment, that is 9:3:3:1 ratio. Okay, but that ratio the Morgan could not get, and it was significantly deviated from Mendel's results. And these are all different results the Morgan could get after crossing or after hybridization between so this kind of characteristics, and with this he could come to a particular conclusion. Now let us see that what conclusion Morgan has got after crossing this particular dihybrid cross after conducting it. See, let us look here. Morgan has conducted the experiment by taking Drosophila melanogaster, and after intercrossing F1 individuals, he got a different idea, different result from the Mendel's usual dihybrid cross experiment. So we have already discussed about the results. Now what Morgan has come to know about this, what conclusion he has come to this after conducting this experiment. Now I can say that when two genes or more genes located or situated on the same chromosome, when two genes, now let us take a chromosome on these two genes, capital A, capital B, two genes, just I am writing genes here, located on the or situated on the same chromosome. So here what happens, the proportion of the parental combination is much higher when it is passed, this chromosome, these genes, when they are passed to the next generation, in the F1 generation or in the F2 generation, the proportion of carrying to the next generation parental combination is much higher than the non-parental combination. Why this proportion of parental combination is higher in the next generation than the non-parental combination, what we call it as a recombination? Why it is higher? He has come to an idea that he argued that, he attributed that is mainly because of a linkage. So here linkage takes place. There is a physical association between these two genes. Because of that, genes could not get separated at the time of a gamete formation, so that as it is they get inherited to the next generation, and this is what he called it as a linkage. Okay, so therefore here what happens, parental combination is higher, much higher than the non-parental combination or recombination. So this is one of the conclusion of T.H. Morgan. And one more thing is, okay, now linkage, what is the strength of a linkage, how linkage takes place? Now say for example, this gene A is present here, close to the A, B is present, next to C gene is present, next to D gene is present, E gene is present. So these are all different locus, different locations, positions of different genes. Here the strength of the linkage is very high between the genes which are present close with each other. And as we move to the next location, next to loci, the strength of the linkage becomes weak. Strength of the linkage becomes weak, so that what happens here, the process of recombination is very, very less when genes are present close with each other. And the recombination chances are more between the genes which are present little bit away from with each other. Now it means between A and B, okay, the linkage is, strength of the linkage is very high, so that recombination chances are lesser. And between A and E, okay, the distance is more, okay, strength of the linkage is weak, so that recombination chances is high. So therefore in his another conclusion, okay, tightly linked genes, so that now these two genes are tightly linked, where they show very low recombination. So there is a less recombination between these two genes. Similarly, when the genes, okay, loosely linked genes, when the genes which are loosely linked, there is a high recombination chances are present here. Loosely linked. Now A and E are loosely linked, A and B are tightly linked, so that in between these two genes, tightly linked genes, okay, very low recombination is present, and between these genes, when they are present apart from each other, okay, there is a distance is more, strength of the linkage becomes weak, okay, less, therefore recombination chances will be more here. So in this way he has come to 2 or 3 conclusions here. And now, so this tightly linked genes showed a very low recombination, and loosely linked genes shows a higher recombination, was explained in another experiment. Okay, here in the genes for white and yellow colors, they showed only 1.3% recombination. It means these two genes are present close with each other on the chromosome. Now let us understand here. Now just I can tell you this. So this is gene for white color and another one is gene for yellow color. These two might be present close with each other. So therefore they showed only 1.3% of recombination. And one more thing is, there is one more gene that is a white color, and the gene which is responsible for a small wings or miniature wings. So there is a one more gene that is a miniature wing. Just I can show with capital M. Now this is white color and miniature wings, and so this gene is present little bit away, therefore the chances of recombination is high here. 37.2% recombination is present. So what we can understand here, the strength of the linkage becomes less here, therefore linkage chances, okay, we can understand with this how recombination takes place. That is what Morgan explained through his different experiments. We can understand about how linkage, what is a linkage and what is a recombination, we can understand with these experiments, and which is very very important for all examinations. Please students understand the concept of this linkage and recombination, and which is very very important from the point of NEET also. Children, look here, one more thing is here, frequency of recombination. You know that very well, recombination is generation of non-parental combination is called as recombination. Now let us understand about the frequency. The frequency of genes depends upon the distance between two genes on the chromosome. The frequency is measured by the distance. I already told you the gene W, gene Y, the distance between the two genes. Based on this we can understand about the frequency of genes. So scientist called as Alfred Sturtevant, a student of Morgan, used the frequency of recombination between genes, gene pairs on the chromosome, and he was the first to measure or map the position of a genes on the chromosomes. Okay, so this genetic map, that is nothing but a position of genetic map, is nothing but the sequence of arrangement of genes. So that genetic maps are very much helpful, and it is used as a starting point, beginning point in the sequencing of a whole genome project in the human genome project. Sequencing of the whole genome in human genome project which is used. So this human genome project you will study in later lessons. So in this way, now let us understand this human genome project in which sequencing is done, and in which so these genetic maps are used as a starting point. Okay, with help of that we can understand about the human genome project and also the genome of other organisms also. So with this now we have understood about the process of a linkage and recombination. Students, please remember, and this is a little bit advanced concept in genetics, and so in examination you will get okay some different concepts, different type of questions in CET also. You please concentrate on this linkage as well as recombination.